Hello, everyone. We're going to do meiosis today. So, what is meiosis? It's different from mitosis because meiosis only happens with sex cells and not body cells. So, the difference is these meiosis occurs in a cell called a gamete. I'm going to spell that right here. Gamete. So, gametes undergo cell division through meiosis, while body cells go through cell division through mitosis. Um, so in human sperm cells, sperm goes through a um, synthesis pathway of spermatogenesis. So how we generate um, sex cells is through spermatogenesis. And this is important because unlike mitosis that goes from two N chromosomes to two N chromosomes, N being 23, 23 from mom, 23 from dad, uh, we go from in meiosis we go from 2n to n and then to n again because you're going through mitosis after you go through meiosis. So to kind of do the breakdown here, we have a diploid, so 2n spermatogonium. Then you go to a diploid primary spermatocyte. This happens in the testes. And then from there, you undergo meiosis one, and then you get the um, haploid's primary spermatocyte, and then the secondary spermatocyte haploid is going to make a spermatid, and that is also a haploid. And then it's going to travel to the epididymis where it will mature into a spermatozoa. And a spermatozoa is a mobile sperm cell that's almost into maturity. It becomes sperm once it ejaculates. But before then, spermatozoa, and it's mobile. It's got the flagella. So on the female counterpart, you have oogenesis. Um, First, you're born with females are born with oogonium in their ovaries, and this primary it goes to a primary oocyte and is arrested in prophase one until puberty. After the menstruation, uh, it is released um, in the form of an ovum, and it is arrested in the uterus is a secondary oocyte and is arrested in metaphase 2 until fertilization. So I'll go over that in um, the phase cycle. So prophase 1, so this would be the primary spermatocyte and the primary oocyte. They're kind of easy to remember because they both are primary. So you start with prophase 1. And similarly to mitosis, they still have prophase, metaphase, anaphase, and telophase. However, there are some important differences. So, first in prophase one, uh, you're gonna have that degeneration of the nucleus again. Um, Everything's normal with the prophase, except this time instead of having two one chromosome getting split up, you're gonna have something called homologous chromosomes, and I've drawn them here. So homologous chromosomes are similar. So they might have a little bit of this DNA, and then you might have one of these. So homologous, it's important to know that homologous means similar. So they might not be the same exact, but they have like similar DNAs. And that's going to be important for something called independent assortment. Independent assortment means that the parental, so when you went from the spermatogonium to the primary spermatocyte, it underwent a cycle of mitosis. And that cycle of mitosis didn't generate two identical cells, chromosomes. So now you have two slightly different but similar homologous chromosomes as you start prophase 1. So now you're starting with 2N because it's still a diploid. So there's two of two. If you look here, we still have the two centromeres. So I'm going to get a nice color for this. Okay, let's do green. Oh, why is that? That's what that's behind here. There you go. So you're going to have centromere here. That's a horrible centromere. You're going to have a centromere here and a centromere here because these are two distinct chromosomes. So there you go. Two homologous chromosomes. They're very similar, but they're different, and that leads to independent assortment. And so in prophase 1, the chromatin is going to condense into these chromosomes, and they're going to sort of pair up with each other. Like, oh, okay, so you're the most similar to me. You have, similar genes that are, the same, but maybe have different expression of genotypes. So, you might have big B, little b, and then this one might have big B, big B. You don't know without, actually genotyping. So that's the independent assortment, and which is important for later. But the key difference from prophase 1 and prophase and mitosis is that you're going to have homologous chromosomes pair up rather than just a chromosome condensing. So you go from 2N to 1N. We're going to have two chromosomes, and then the daughter cells are going to have one chromosome each. So, like usual, the nucleolus disappears, the nuclear envelope disappears, the microtubule central organization centers start to appear, and they form from the centrosome. This is all review. Um, you have the polar going towards the chromosomes, and you have the astromicrotubules behind. So here's a little drawing. See, they're kind of paired up, but not in the center yet. Hey, we have similar genetic code. We're homologous. Let's get together. So this is prophase 1. Then you have metaphase 1. So metaphase 1 is similar in the way that the chromosomes line up. However, it's different because now you have two homologous chromosomes instead of one chromosome. So it can't go on the center. So instead, they split the center line. You still have the sister chromatids of each chromosome, but the homologous chromosomes are going to line up at the center, and they're going to split it. So the homologous chromosomes pair up at the metaphase plate, I should say. And the microtubule organization center migrates to the poles of the cell. And the polar microtubules begin reaching out and connecting to the kinetochore. This is our view, and they hook on to the kinetochore. And so the microtubules connect to the kinetochore on each of the homologous chromosomes centromere. Center. Central. So there you go. See here from this Pablo Picasso publication of art right here, you see that we have the microtubules, they're reaching out, the polar ones, they're reaching and they're connecting on the kinetochore of each of the distinct homologous chromosomes. They're similar, but they're not the same. Oh, one thing I forgot to mention, which is kind of bad. Yeah, that's kind of bad. So in prophase 1, you know how I said they were pairing up with each other? So in that process, they undergo something called crossing over, which is actually massive. I forgot that. Crossing over. And in this process, that's why I drew these. They are not the same due to independent assortment, as I mentioned earlier, since the spermatogonium underwent mitosis. They're not the same. but they're very similar. And then in addition to that, you're going to have crossing over, which involves um, this chromosome is going to overlap and take some of that. And this chromosome is going to overlap and take some of this chromosome. So then the results of those two chromosomes, let me draw it again real quick. Oh, excuse me. Thank God. Okay. The result of those two chromosomes crossing over are going to be um, the result of those chromosomes crossing over is going to lead to this one that had the blue, and the blue just symbolizes some gene expression, is going to be somewhere over here, because the blue, you know, it was native to this chromosome and it crossed over. So they like overlap. They go like this. And while they're doing this, they're exchanging genetic information. Um, that's the best way I can explain it. So this homologous chromosome gets some of this homologous chromosome's DNA, and this homologous chromosome takes some of this homologous chromosome's DNA. That's why kids be looking different, um, even though they come from the same parent, because of independent assortment and crossing over. That happens. Independent assortment happens in the mitosis phase prior to meiosis one, and the crossing over occurs um, during prophase one when they're pairing up and finding out which chromosomes are most similar to them. So now that we've addressed that, we can move forward. Um, so the homologous chromosomes pair up at the metaphase plate, and they migrate to the poles of the cells. This is our review. Anaphase 1. So now we get to the part where they're separating. So now we see these homologous chromosomes actually being pulled to the other side of the cell. So this cell, as of right now, is 2N because there's still two sets of chromosomes in in this cell. So there's this set and there's this set, and they all have centromeres. And if you look at the mitosis video, you'll know about the anaphase promoting complex, the checkpoints, the motor proteins that use the microtubules in the mitotic spindle to migrate to opposite poles of the cell. But we're not going to review that here. This is just highlighting the differences between meiosis and mitosis. So after the homologous chromosomes have separated in meiosis 1, telophase forms the cleavage furrow, the myosin and actin proteins, which form the infrastructure, and the intermediate filaments, I think, are also involved in this. They're going to form a cleavage furrow, and the nuclear envelope is going to reform. After the nuclear envelope and the nucleus reforms, cytokinesis separates the cytoplasm organelles and N chromosomes, because now we started with two N homologous chromosomes, right? And each cell got an N. So now these two cells are both these two cells are both N. We took 2N and we made it 1N, because each daughter cell got one set of chromosomes. So then after that, so that sums up meiosis 1. It's going to be followed by, the telophase, the G0 phase, the quiescent phase, and the resting phase, where it's going to require a stimulus to go into meiosis 2. And we know that meiosis 2 is very similar to mitosis, besides the fact that you're only using N chromosomes rather than 2. So that's just the main difference here. It's just like, okay, now you have all these haploid cells, and in the oogenesis pathway, when the oocyte undergoes meiosis one of those oocytes is known as something called a polar body. Um, it's not going to get all of the DNA. So one of them, one of these, is just going to like literally just get bombed. It doesn't get any. It doesn't get any DNA, and that's just an L. So yeah, that's just important to know. And it's called a polar body, and it occurs in um, female um, gametes. So after it undergoes meiosis, uh, it becomes something called a polar body. You can look that up if you want. I go and do it too much here, um, but that polar body just gets excreted. It just degenerates because it can't sustain itself. It doesn't have any DNA. So then it's senescent, and then it goes to prophase two. So prophase two, um, we know that uh, this is where the secondary oocyte, um, the secondary spermatocyte is going to undergo this. And the secondary oocyte and the secondary spermatocyte, the chromatin is going to condense in prophase 2, like normal. You're going to get the U chromatin to the heterochromatin, U meaning same, hetero meaning different. Well, I don't know if U actually means same, but that's how I kind of reason it. It's going to condense, the nucleus is going to disappear, the nuclear envelope is going to disappear, and microtubule or centriole organization center, microtubule organization center. Excuse me. Good God, I understand this. Microtubule organization center is going to form from the centrosome. Um, polar and astro microtubules form. Same old, same old. The key difference to know here is that this cell, when it's undergoing meiosis 2, you should automatically think haploid. This is a haploid cell. It only has N. Diploid means 2, hap. Well, I'll let you draw your own conclusions there. Um, so then we progress to metaphase 2. Now in metaphase 1, we had homologous chromosomes and they split the metaphase plate. In metaphase 2, we only have one chromosome per, we don't have homologous chromosomes. We have sister chromatids connected at a centromere. Um, so just like really just cement those concepts into your head. Just like homologous chromosomes, oh, meiosis 1. Sister chromatid, meiosis 2. So in metaphase 2, they line up at the sister chromatids. Each chromosome lines up, excuse me, at the metaphase plate, and the mitotic or microtubule organization center migrates to poles of the cell. Polar microtubules connect to the kinetochore on hook on the centromere on each side, just like meiosis or mitosis. Excuse me. Then, like normal, oops, just like normal, you're gonna have those cells latch onto the kinetochore, which is attached to the centromere. You're gonna have that securin removed by separase, and then that cohesin is going to degrade by separase, and it's going to pull it. Well, it's still metaphase, so we're getting ahead of ourselves. But the sister chromatids are now ready to be separated, and we know now that we only have N, so we have a haploid number of chromosomes, and they're going to split. They're going to be lined up on the metaphase plate because they're going to split down the centromere. It's also important to note that the secondary oocyte is arrested here. So while it's chilling in the, the uterus, or that uterine lining, and it's waiting to be fertilized, this is where it stays. It's like, all right, I'm waiting, and I'm on the metaphase plate. Uh, Now I'm either going to get those extra chromosomes, or we're going to excrete this. So If it doesn't get it, if it doesn't get fertilized, then the secondary oocyte goes uh, through. I think that's menstruation um, or a period. If it does get fertilized, and we have a little sperm guy here, this would probably be a spermatozoa, but it's been ejaculated, so now it's a sperm. Penetrates that um, secondary oocyte, adds that second set of chromosomes. Now you went from haploid to now this is diploid. And that is classified as a zygote, as I've written very largely here. So a zygote is a successful fertilization event, and that leads to 2N. And now that is going to grow on to become life, which is kind of crazy, through several mitosis cycles. Um, and that's all like really fascinating stuff, how it happens, how you go from like the blastula to the blastocyst to the morula, blastula, blastocyst. You get the trophoblast. Like it's a pretty in depth kind of thing, but it's really cool. You start seeing like the nervous system forming and like your skin. But anyways, I digress. So after um, metaphase two in the secondary spermatocyte, you're gonna have anaphase two, where the motor proteins, kinesin, dynein, etc., pull the chromosomes, pull the sister chromatids here. I gotta be better with my English here. Sister chromatids, because remember, it's haploid. The spermatocyte is haploid. So essentially, um, yeah, I'm not quite sure if the um, secondary oocyte, once fertilized, I think it goes through mitosis because it becomes diploid. Um, or it might continue with anaphase two and just separate those two and, do the whole crossing over event. Um, but that's way too specific. We're just going for the general um, gist of meiosis two. What you need to know is that the sister chromatids of the secondary spermatocyte um, are separated, and now you have four. Um, uh, well, you're gonna have four. So two cells are doing this. Two haploid cells are doing this, and the sister chromatids separate to either side of the cell, just like in mitosis. Telophase, you're gonna have the pinched myosin and actin proteins forming that cleavage furrow, and then cytokinesis occurs, separates the cytoplasm organelles um, excuse me, and chromosomes equally. So that now you have in total from meiosis one and mitosis or meiosis two, you're gonna have um, four. Because remember, we had two cells undergoing meiosis two, um, and each one of these is gonna have one sister chromatid, and they're all going to be different, which is crazy. So you have basically a 1 in 4 chance of getting any gene, but then you got to get into genetics, and you got homozygous dominant, you got heterozygous, you got recessive traits, and the ratios. If you know anything about Gregor Mendel, yeah, if you want to know more about that, I'd look into him. Um, but yeah, then it's telophase followed by the resting phase, and it's pretty stable. Um, so at this phase, the spermatocyte, secondary spermatocyte, will go to the it'll become a spermatid. Jesus, excuse me, spermatid, and then uh, it'll go to the epididymis to mature into a spermatozoa. There we go. And that's where it has the flagella. And that spermatozoa, it's going to become a sperm once ejaculated and do this, meet up with the secondary oocyte. They both have N chromosomes. They're going to insert the genetic DNA, create that zygote, and that zygote is going to undergo mitosis because it's no longer a gamete, it's a zygote. And zygotes undergo mitosis. So the homologous chromosomes are going to get separated, and now both, well, they're going to replicate. So it's going to be essentially foreign. Once it's synthesized, it's got to go through the whole mitosis phase, interphase again, too. So the zygote's going to take those two homologous sets of chromosomes, that homologous set of chromosomes, 2N, and it's going to replicate them to 4N before undergoing mitosis. So, yeah. And then the morula, the blastula, blastocyst, trophoblasts, goodness, gastrula. And that whole process, like if you want to know what that process is called, it's gastrulation. It's really interesting. So you're gonna have a bunch of signaling molecules because those first, like that zygote, once it forms that morula, they're gonna have like very specific cells because they need to make a whole human from one cell, the zygote. So that process is called gastrulation. If you're interested in that, I definitely look it up. There's probably something on YouTube that goes over it. It's fascinating. And if you look at it, don't try to like dig too deep into it. Like unless you're interested in it, like go for it. Be my guest. But it's just cool to have an understanding of how we are created from one single cell. Yeah, like that's crazy. Like omnipotent stem cells are wild, um, and there's different kinds. Uh, but so that's meiosis in a nutshell. I hope you enjoyed. The next video we're going to do is some laboratory techniques like flow cytometry, Western blotting, um, genotyping, uh, or polymerase chain reaction, um, QR quantitative reverse transcriptase polymerase chain reaction. We'll go over some gel electrophoresis techniques and immunofluorescence. I think those are going to be the main ones we'll go over, just so that when we finally start going over these papers, we can like critique their techniques and be like, hey, is this the best way to look at this, or is there a better way? And if so, like, what do you think the message they were trying to send with this figure is? And then we can start like really seeing what advancements in these papers are actually worth following up on and what to look out for in the future. And then what could have been done better, because like we should never stop improving as like scientists and in any subject, like not even just science. Like if you're if you should never stop getting better. So there's always something. um, Main takeaway from meiosis is that the result is 4 haploid um, gametes that are all different. um, they due to independent assortment and crossing over. And then, of course, um, the primary oocyte is um, arrested at prophase 1 until puberty, and then the secondary oocyte is arrested at metaphase 2 until fertilization. Because so when people say it's really up to women, like women hold the key to like life because they're literally waiting on men, like even at the molecular bio level. So it's it's interesting if you like it. It is what it is. Look more into gastrulation, and I hope you enjoyed this video. And see you next time.